This is the genetics lab, and we're going to do there's a genetics 1, there's a genetics 2, and a genetics 3 handout as well. And they're basically, there's information about genetics as well as how to do some of these Punnett squares. And then you could do them yourself, and I encourage you to do so. But I'm going to go through most of the genetics 1 handout today. I'm not going to do every single problem, but I'll do a few of them. And one of the things you might see when you take the practical is this image of called the Monohybrid cross, and it's from corn, right? And so this is corn is actually a seed, right? You plant it, it makes a corn plant. But these are two different phenotypes that corn can have, right? These are called colored, and these are colorless. Or I could say that's purple and that's yellow. That's another way of describing it, But on the box, you got boxes on your bench, right? They use the term like this is the parental P1, which we covered in class. So when they cross this corn and this corn, it developed the F1 generation. In the F1 generation, all the corn is purple or colored. That means it's dominant over the yellow color. When they cross the F1s, and they had baby corn, then the corn had a mixture. Some of the seeds shows the recessive phenotype, but most of the seeds or kernels show the dominant phenotype, right? But that's what this is, right? It's a series of generations, right? You'd say it's actually, like if I use big A, that would be the dominant phenotype or allele. These guys are little a, or the recessive allele. Now they use it's hard to tell the difference between this C and this C. This is an uppercase C. This is a lowercase c. And then they show two of these, which just means it's they're crossing this with this, but they're both the same genotypes. They're heterozygous, big C, little c. That's their genotypes. So the genotype are letters. It gives you the definition of an allele. That's a single gene, but it can have two variations in this case. Sometimes there's three, but usually it's just two. Most of the time, there's no variation. Phenotype is the visible or observable characteristic. So you would expect then, in this cross, right, big A, little a, big A, little a. If you do that cross, then you get a Punnett square. This is the Monohybrid cross, right? And you get this guy generates these gametes. This guy generates these two different gametes. And then if this gamete and this gamete hook up, you get the big A. Big A, that's the genotype result of that cross. If the big A and little a hook up, right, so this guy, this is the F1. It's similar to what happens here. We're crossing this guy and this guy. It's using big C and little c for that, but we're using big A and little a, big A, little. It's heterozygous. They're purple or colored. Then these two are the gametes that this guy generates. 2N, this is the 1N state, right? This say is sperm, and then these say are the eggs that are 1N. When this sperm and this egg unite, you get that genotype and that offspring. What's the phenotypic ratio of the F2 corn? It's 3 purple or colored to 1 colorless or yellow. But I might ask you that, what's the phenotypic ratios in the F2? What's the phenotypic ratio of the F1? They're all purple or colored. And at the bottom of the box, you can see, this is the phenotype. Purple alluron, colorless alluron. You can just put purple and colorless for your answer. 3 purple to 1 colorless. These are the genotypes here. They're not telling you what the phenotype. If you put genotypes when I'm asking for phenotypes, it's wrong. Now we describe this as being a dominant trait. So you have dominant recessive traits. Then we'll talk about the incomplete dominance trait. And then there's even a codominant trait, right? But you should understand those distinctions when you take the practical or exam 4. Like the big A, little a, the explanations of them. So if you just follow those directions, it reemphasizes the difference between the phenotype and genotypes. Right, you should understand if it's a capital letter, it represents a dominant allele. If it's a lowercase, then it's a recessive allele. Now, in this depiction, right, they're going through purple. Instead of yellow we used before, they're using white. Right, so the dominant homozygous or heterozygous gives you the dominant phenotype. And then here's an example of a question about, and how you would fill out. The cross here would be, I don't know if it's real clear, but it's big A, little a is the cross. Right, for that particular question, it's asking if you have this cross, how would you complete the Punnett square? Big A goes there, little a. Half of the gametes have this, the other half have that. It's the same allele, but one's from one parent, one comes from the other parent. But you notice in that cross, they're all heterozygous, the result. That's why you say the F1, they're all the same genotype. That means they'll all have the same phenotype too, right? They'll all be the dominant phenotype. So like they even what's nice about this is then it's saying, if you were asked the question, what are the genotypic frequencies for this? They're all little, big A, little a. Or in this case, there are 4 of them, 0 anything else, right? But what's the phenotypes? All 4 are purple. So you could use the word all purple or 4 purple, either way. You count out the squares to get the exact numbers. If it's 3 to 1, that makes more sense. Or, 1, 2, 1. And so in this question, right there, they're asking you to do this cross, big A, little a, with a white flower P, which would be little a genotype, right? This would be the cross. And then it's asking you to fill in the Punnett square for that cross. And then fill in what the genotypic ratios would be and what the phenotypic ratios would be for that. Right, so if we did that cross, it'd be, say, little a from this guy, and then big A, little a for that guy, and then that would give you, right, this Punnett square. You'd have the genotypic ratio would be 2 big A, little a's, to 2 little a little a. That's the genotypic frequency. The phenotypic would be 2 purple to 2 white. But that's what that's asking you to do. And just by going through it, it kind of helps you learn the difference between phenotype and genotypes. And then I already did this cross already, right? Big A, little a. That's the heterozygous cross, where you have, this is a monohybrid heterozygous cross, and you get the typical 3 right, you get 3 are going to be purple, 1's white, and then the genotypic ratio is 1 big A, 2 big A little a, and 1 little a little a. Right? So the number of the squares go for each of the genotypes. The genotype ratio would be different than the phenotypic ratio in this case. But that's then there's one about dwarf there. And then this one is about, they're comparing, it's not this simple, but in this scenario they're making it simple. If you have the brown eye, it's dominant, you get the big B. So that means, right, if you're big B, or big B, little b, that gives you brown eyes. And if you're little b, that gives you blue. Running out of chalk. Now, if you look at this, you can figure out what if they're blue-eyed, all of them, you know that they have to be, right, little b. So that means this guy has to get the little b from mom, the big B from dad, and he has brown eyes. So you know what that genotype is. If these two are brown and this one's blue, then both of these guys, this guy has to have the little b, this guy has to have the little b. And that's how you end up. So you should be able to go backwards. It'd be nice if you could. So then if you have an incomplete dominance, which I'm glad we already covered that in class, then what that means, if you're heterozygous, we don't use big A and little a, right? We could say we're going to use R, CR, and CW. So if you're C big R, C big R, then you get the red phenotype. If you're heterozygous like that state, then you get what's called, you get a pink instead of red. And then if you're CRW, you get the white. Yeah, homozygous means it's the same allele, right? These two are homozygous. In the heterozygous state, you see neither red or white. So then you say it's an incomplete dominance. You get in between, or a blending is another way of putting it, right? It's both white and red are expressed. That's why it's pink. There's other things. You could also say, let's just say instead of, you get the long tail if you're homozygous, and then you get the short tail if you're homozygous for the other, and then if you're heterozygous, then your tail is medium, long, medium, and short. So it could be length, or it could be a shape. Could be, there's a radish question, and there's a long radish, there's a round radish, and there's an oval radish. Same scenario. Instead of colors, it could be shapes or lengths. Okay? It's a blending or an in-betweenness, it's called. That describes incomplete dominance. But you don't see in the heterozygous state, because we normally start, when we do the crosses, we start with, right, the heterozygotes. Right, we say red and white, and that are homozygous states, and that generates in the F1 the heterozygous state. So the F1 will tell you what you want to know about whether it's incomplete dominance or if it's dominant. If it's in the heterozygous state or in the F1, if you don't see red or white, then it's incomplete. Then this would be, right, the first, this cross here that I put on the board is what, and this would be the Punnett square. You're not going to have to do a Punnett square like this for the practical, so, just doing it, I think, helps you understand. If you want to know the genotypic ratios here, all of them are the same, CR, CW. The phenotypic ratios of this, they're all pink. They all have the same color, right? But you don't want to, even as simple as this looks, the more familiar you are with it, the more successful you'll be. I don't think you're going to need to do it. Then a pink flower, and a pink flower would be CR, CW, CR, CW, and then you fill it in. Now, let me show you on the PowerPoint, we have both of those examples, right? Here's the monohybrid. This is from your textbook, right? You have red, white. Here's the genotypes. I put them on the board. F1, they're all pink. This is the Punnett square for a cross of this generation, right? Two of these generates this cross. This would be the offspring. The F2 generation would have these genotypic frequencies and phenotypic frequencies. Now, in this case, the phenotypic frequencies and the genotypic frequencies are the same, right? You have one red, two pink, one white for phenotype. You have one CR, two CRCWs to one CWCW. So the genotypes and the phenotypic ratios are 1 to 1. So they're the same. But that's incomplete from the textbook. And then I've also on the PowerPoint, I've got, this other, right, this is a monohybrid cross. When you cross this heterozygote with another heterozygote, that is the result of that cross. And then here's a better picture of that monohybrid. It's on the handout. This is P. I've got purple, yellow. What's the phenotypic ratios of the F1 generation? It's 3 purple to 1 yellow. What's the phenotypic ratio of the F1 generation? They're all purple. The F1 is here, F2 is this guy, and that's the P1. If you're doing a dihybrid, then you get, I don't have it on here, I don't think. The dihybrid like this, you get a 9:3:3:1 ratio for the phenotypes. So those are some oval questions about that. I told you about the rashes. Now a multiple allele is a little bit different, and you don't have two outcomes. You can have, in this case, there are 3, right? So the 3 alleles are, let's just call this A, and we'll call this one B, but you don't use A and B I asked you about that on a test, right, it's blood type A and blood type B, blood type AB, and blood type O. Those are the phenotypes. The little I is recessive to these two in a sense. But you refer to this as it's multiple alleles, so there's two descriptions. There's more than one, or more than two rather. This one, or this one. And then it's codominant. And a codominant is when this scenario here, where you have two different alleles, but they both show up on your red blood cell, right? If you're AB, then on your red blood cell you have both of these antigens, they're called. If you're blood type O, then you have little I, you have neither A or B alleles, and you'll have your blood type is O. So if you're told someone's blood type, if it's O, then you know this is their genotype, there's only one. If you're given whether they're blood type AB, then you know this is their genotype, and that's what's used for that. But you have two different genotypes if you're blood type A and B. If you have this gene, then that's what you get on your red blood cell. If you have this gene, you get the B. If you have neither of them, you don't have A or B. So this would be how you would do a Punnett square with someone that's AB with another person that has blood type AB, right? These would be the alleles for one person, the other person. Then when you fill in the Punnett square, this person would have blood type A, this person would have blood type B, right? And then two of them would have AB blood type. And then down at the bottom, if you look, right, they're written the ratios, the genotypic ratios. It has to be written with the I's. That's how you abbreviate the allele. These are the blood types. So it's okay to use A, AB, and B for that. I mean, technically you probably should say type A and type AB and type B. That would be the most correct, but we're kind of leaving them off. But see, notice the ratio for the phenotype is the same as the ratio for the genotype, right? The numbers are the same. Let's get, the last thing I want to cover is this dihybrid scenario. So in dihybrid, we're looking at two genes, right? So let's say the A gene is here, and the B gene is another gene. If you do a cross with this guy and this guy, then they're all going to be heterozygous, right, from what we did before. Then if we add the B gene and use the little b and the big B, right, these are homozygous, recessive, homozygous for two dominant. Then in the F1, this is what results, the heterozygous state. So a dihybrid heterozygous cross is when you do the F1 cross, which is they're both heterozygous. Right, that's what this is attempting to explain. So in this cross, you generate 16 squares that you see here. And to get the gametes, this guy this individual and this individual, they both generate the same gametes, right? They're shown here. And the way you get them is you say, this guy hooks up with that guy. That gives you the big A, big B. And then big A hooks up with the little b. That gives you that genotype. And then the little a hooks up with the big B. That gives you that genotype. And then the little a goes with the big B. All right. This guy does the same thing and say that's what generates these gametes. When you fill in this square, right, you get big A, big A, big B, big B. Right? The next square you would get big A, big A, big B, little b. That would go in this square. Now this square, you get all of the, you get little a, little a, little b, little b. One of 16 squares are going to be little a, little b, and that means they show both recessive phenotypes. And I explained in class that 9 of the 16 squares, or 9/16, show both dominant phenotypes. So 9/16 and 1/16 are the two important ones. Both recessive, both dominant, when we're referring to a dihybrid heterozygous cross. It doesn't matter what the phenotypes are, right? That doesn't, it can be anything. As long as it's dominant recessive, we're talking about 2 genes instead of 1. And then I did the trihybrid in class, right? If I said if there's 3 genes, A, B, and C, then you'd have a 64 square Punnett square. Now, if you kind of get what all that means, that's great. If you don't, it's no big thing. You know what I mean? If some people get it, that's great.
Now is that?
And now this is the Dihybrid Cross on the lab PowerPoint. This is the, we have boxes of corn in the lab, and they show corn on the cob. It's got, this is the parental generation, which generates the F1 generation. This is typically shown in biology labs. They use the colored alluron for this purple color, I guess you could call it, and then colorless for this yellow color. Right. And that's one phenotype, right? With the dihybrid cross, there's 2 phenotypes or 2 genes. Okay. One gene is, you could say it's the R gene, the other gene is the SU gene. That's kind of complicated. You could just think of it as the A and the B gene as well. It doesn't matter what the letters are used. It's just, if it's upper lowercase is the important part. So I just want you to understand what this means underneath the term, right? These are the genotypes below the corn. Okay, this is not the phenotype listed here. Okay, the phenotypes are over here and an explanation of them just like I have over there. So you say this is dominant over these 2 phenotypes or traits, because when you cross these 2, the dominant traits are what you see in the F1. It's no different than parental F1 that we've talked about in class or before. It generates, when you cross the F1, the F2 generates a 16 square Punnett square. I think I told you, right, that in this one square, 1 and 16 of the offspring show both recessive phenotypes, right? They show this particular corn, right? It looks wrinkled is a nice way of putting it, I think. This is round. Okay, so if I asked you what are the phenotypic ratios of the F1, right? They are all round and colored or purple, right? It doesn't matter if you use purple or round, purple or colored, either way I'd be okay with that. And the same thing, round and wrinkled could be used, or if you wanted to use it's starchy and versus sweet endodermis, the way they describe the phenotype, but it's not important, you know, what you put on the test. Now, this is the whole box here, right? The F1, this is the parental F1, then this is the F2. Now, the corn you see here in the F2 can have, there's going to be 9/16 of the corn in the F2 are going to be purple and round and show both dominant phenotypes. Right, so if you look, you could even count them and see some are yellow, right? Some are round, some are wrinkled. 1 and 16 in the F2 are going to show both recessive phenotypes. Okay, so the 9/16, they have 9 here. When they show this genotype here, it's for the phenotype. Okay, 3 are going to have, say, purple and wrinkled, 1 dominant, 1 recessive. The other 3 are going to be round and yellow. And then 1/16, 1 out of the 16 are going to be both yellow and wrinkled. So I just wanted you to understand what the corn does here, because you'll get questions about it on the practical. And I expect you to know, right? It's no different, right? Remember, the letters aren't critical that are used. It's whether they're upper and lower case is important. I would probably stick to the phenotypes that I've listed here because they're a little bit simpler, but you don't have to when you do the test. So that's the dihybrid. I wanted to kind of finish this video up with that, right? In class, this is equivalent, like I put it here, right? This is equivalent to what you see there, right? They're just using Y and R instead of A and B. If you don't quite get that, that's not that important. So, anyway.